It's my pleasure to introduce our first speaker, Shabih Shakeel. Um, he's a chromatin researcher that has been, throughout his career, doing some really outstanding work using structural biology to understand the way that biology happens. He started his graduate training doing his PhD with Sarah Butcher at the University of Helsinki in Finland, where he solved some really cool structures of picoRNA viruses or picornaviruses. I'm not sure which is which. Following a brief postdoc still in Helsinki, he then became an MRC Career Development Fellow in the United Kingdom at Cambridge, where he worked with Laurie Passmore and found some amazing structures and did some amazing work looking at the Fanconi anemia core complex. And after that, he transitioned to his current institute at the University of Melbourne, where he is a professor and he is studying the structure and the function of the MORC2 chromatin remodeling complex. And today he's going to tell us about his findings. So, Shabih, please take it away.
Thank you, Jackson, for the kind introduction and for accommodating me in this order. So I'm joining from bright Melbourne. It's 7 in the morning here. So let's get started. So today I'm gonna speak to you about the mechanisms of chromatin remodeling, which take place by MORC family ATPases. So I don't have to preach to this audience, but chromatin remodelers, they govern various cellular functions, ranging from DNA replication all the way to transcription and DNA repair. And the way they do it is by controlling accessibility to the DNA. There are four well-characterized families of chromatin remodelers known so far, and all of them have the common feature of containing an ATPase domain within them in one of their subunits. And that's why MORC is also similar, because it does contain an ATPase domain within its protein, and it does do remodeling as well. So there are four different types of human MORCs. They are all GH-kill type ATPases. So they are MORC 1, 2, 3, and 4. So the GH-kill type ATPase domain is on the followed by coil domains, and then zingers, and then there is the C-terminus, which again contains some coils. Among all these four MORCs, it's the MORC2 which has got the highest disease burden in it. It is overexpressed in a variety of cancers, and it's also involved in neurological disorders. And the two most well-studied are the Charcot-Marie-Tooth disease and spinal muscular atrophy. And there are already several patient mutations identified within MORC2 leads to this disease. So one of the famous cases of Charcot-Marie-Tooth disease caused by MORC2 is within the Samsung family, which makes all our phones, fridges, and TVs. And it was known recently that the family was suffering from it, and they have got some good news that there are some therapies, gene-based therapies, which have helped this family to overcome. So the main function of MORC2 is epigenetic silencing, and the way it does it is that it's a part of a bigger complex called as HUSH, where the HUSH comes and reads the H3K9 trimethylation marks on our chromatin. It then recruits a complex of methyltransferase ZTB1 together with its interactor ATF7IP. That eventually leads to the recruitment of MORC2, and it's believed that once the MORC2 is recruited, then heterochromatization of that particular region happens, and that leads to epigenetic silencing. Besides that, MORC2 also plays a role in DNA damage, as has been shown by a few papers recently. And the interesting bit is among all these papers, it's actually the function of the MORC2 is attributed to its post-translation modifications. However, even given the importance of MORC2 in different disease types, there is a lack of understanding of how MORC2 functions, especially at the molecular level. So we don't really know how MORC2 is regulating epigenetic silencing. We don't know how post-translation modifications play a role in DNA damage, and maybe whether it has other roles as well, and whether there is a switch where MORC2, instead of doing its epigenetic role, it switches to a DNA damage response. So that cue is actually missing. So my lab has taken a reductionist approach where we try to understand the function of MORC2 in a test tube. So we do in vitro reconstitution of MORC2. And here I'm showing you the full length MORC2 expressed in insect cell expression system. And we show that MORC2 indeed contains the ATPase activity as shown here. And it's not really influenced if the DNA is present or absent in order to impart the ATPase activity of MORC2. But more importantly, what this shows you is that the recombinant full-length MORC2 is indeed functionally active. So now we can go ahead and do some assays. So the first one we wanted to understand was the oligomerization state of MORC2. To do that, we first expressed and purified just the ATPase domain of it, and it shows up as 69 kilodalton as expected. If we give it a non-hydrolyzable form of ATP, it does dimerize and it shows up as 144 kilodalton. In comparison, if we express and do the size exclusion chromatography coupled to moles, we see that MORC2 wild type itself is coming down as a dimer. And we believe that dimerization is through the C-terminal domain. Now, also to prove that, we used a technique called as fluorescence fluctuation spectroscopy. That work was done in collaboration with Liz Hines' lab. And what we did there comparison between a monomeric GFP within cells as compared to MORC2, which is fused to the monomeric GFP. And by looking at the diffusion rate of how MORC2 is diffusing in the cells, we identified that about 10% of MORC2 comes down as a dimer, and that actually increases when there is a DNA damage happening. We then confirmed the dimerization state of MORC2 by using atomic force microscopy, and that gives us a direct visualization results showing that MORC2 indeed is dimerizing, and it's dimerizing through its C-terminal domain. We again wanted to understand whether when we introduce the AMPNP, like in the previous slide, is it going to lead to the dimerization of its ATPase domain, which is on the N-terminus, and that indeed does happen. So now we have got a visual proof showing that MORC2 is a dimer through its C-terminal domain, and it also dimerizes through its N-terminus domain when there is ATP or non-hydrolyzable form of ATP. And this atomic force microscopy was done in collaboration with Jay Kyun-Ryu's lab in Seoul National University. We then went on to solve the structure of full-length MORC2. Within our crime structure, we can see two copies of MORC2, but only for the ATPase domains. So the C-terminal domain remains highly flexible. That tells you that flexibility is important to impart a function for the MORC2. But more importantly, we were able to reach about 2.5 angstrom resolution for this full-length MORC2 map, and that also can in future give a structural basis if someone wants to design rationally designed drugs against it. So there is a structure, and then if you come up with like few good drugs, you can actually go back and with structure at high resolution to understand how those drugs are gonna affect the MORC2. That's all very exciting, but we wanted to probe MORC2 function further. And now we wanted to concentrate on seeing the effect of post-translation modification. And in this regard, we first concentrated on phosphorylation, and we observed that MOC2 is indeed heavily phosphorylated in its C-terminal domain around the coil 3. And these phosphorylation sites are actually very well conserved, and we have now coined a term PIM domain for this particular region of MOC2. And interestingly, the MOC2 is coming phosphorylated from like the insect cells. So we don't really have to give it any extra kinases or anything in order to cause phosphorylation, which is quite interesting. So we wanted to probe what happens to MOC2 when it's phosphorylated. So first, we did an ATPase assay where we had the wild type MOC2, which is already coming phosphorylated, and show that yes, it hydrolyzes ATP, and the hydrolysis of ATP between the wild type as compared just to the ATPase domain, which is shown here in the middle as 1 to 603. The kcat value is very similar. However, if we take a phosphodiesterase version of MOC2, where all the phosphorylation sites have been converted into alanine, we do see that the rate of hydrolysis has doubled, and that shows that dephosphorylated MOC2 hydrolyzes ATP at much higher rates. Would that also translate into its binding ability to the DNA? So for that, we took help of surface plasma resonance, and we showed that the wild type and the ATPase domain have very similar kind of binding affinities. However, the phosphodiesterase version of MOC2 binds almost 5 times with stronger affinity as compared to the wild type and the ATPase domain. So that shows that dephosphorylated MOC2 indeed binds with much higher affinity and also has like much higher rate of ATP hydrolysis. We then wanted to probe within the cells like where MOC2 is actually the regions within the genome. And for that, we collaborated with Resilence Lab and Gordon Smythe's lab at VHI, and using the ChIP-seq analysis as well as ATAC-seq analysis, we showed that MOC2, although it's meant to work together with Hush, which recognizes the H3K9 trimethylation marks, here we see an anti-correlation where the 2 is present wherever the trimethylation mark is not. And if we look at it a bit further, we see that most of the MOC2 is actually within the promoter regions of the genome. And then the next one is within these line elements, which are known to contain the H3K9 trimethylation marks in them. This was so we wanted to confirm it with an orthogonal. So for that, again, we collaborated with Liz Hines' lab, and using FLIMFRAT coupled to immunofluorescence of either the MOC2 or of the H3K9 trimethylation mark, we showed that indeed MOC2 is binding to the open chromatin regions, which could be attributed to the promoter regions. Next, we wanted to understand how MOC2 undergoes, whether MOC2 undergoes any kind of structural rearrangements in DNA. So for that, we quantitative cross-linking mass spec, where we used a high-density cross-linker called as sulfo-SDA, and we conducted an experiment where we have either MOC2 by itself or in presence of DNA. When we mapped those cross-linking regions on the MOC2 domain organization, we can see there were subtle structural changes which were happening. But when we mapped it onto the structure, we can see that within the 2 protomers of a MOC2 dimer, there were a lot of cross-links between the 2 protomers in the absence of DNA. However, the moment you add the DNA, those crosslinks disappear. So what this tells you is the DNA binding is somehow relaxing that dimer of MOC2 or making it more flexible, and probably that's important for its role, which is implicated in chromatin remodeling. We then wanted to probe whether MOC2 has got multiple DNA sites on it or not. So in order to do that, we took help of hydrogen deuterium exchange mass spec, where we did the mass spec in presence or absence of the DNA. And we observed that in the presence of the DNA, we can see the DNA binds strongly in these blue regions, which are within the coil 1. And those are like well-known sites for binding of MOC2 to the DNA. However, we also found sites belonging to the zinc finger, but even more interestingly, when we took the C-terminal domain of MOC2, we also started seeing DNA binding sites within the C-terminal domain, which was quite new. So this work was a collaboration with Sheena and Sheena Darcy at the University of Texas, Dallas. And based on these observations coming from Sheena's lab, we performed AMSAs using many different constructs of MOC2. And in a nutshell, what we see is that indeed there is binding ability of the C-terminal domain of MOC2, and then all of these different domains have got like different affinities for the MOC2. So what it tells you is that MOC2 has got multiple DNA sites spread all across it, and all of them have like different affinities. We then understand how MOC2 engages the DNA. In order to do so, we performed an electrophoretic mobility gel shift assay where we had where we wanted to see whether we give the DNA to MOC2, is it gonna clamp on it or not. To do so, we took the help of those AMSA gels and using a DNA competition assay. And what we did was we had one form of MOC2 which was not pre-incubated with the DNA. Then the second one was pre-incubated with linear DNA. And then the third one where it was pre-incubated with the circular DNA. Once that pre-incubation was over, we tried to compete out using a linear form of DNA of similar length, and we run AMSA gels for that. So long story short, what we observed for the full-length MOC2 was it's easier to compete out, excuse me. So what we observed was that it was easier to compete out the linear when the pre-incubation happens with the linear DNA, but it was much more difficult when the pre-incubation was a circular DNA and then the linear DNA was not able to compete it out. In comparison, what we observe for, if we just take the ATPase domain by itself and do the same experiment, we see that both in cases of circular DNA and the linear DNA, it can easily compete out the pre-incubated DNA. So what it tells you is that MOC2 indeed clamps onto the DNA, and that clamping happens through its C-terminal domain. Now, in order to prove our biochemical observation, we also did atomic force microscopy again in collaboration with Jay Kuhn's lab, and we indeed showed that MOC2 is engaging on the DNA through its N-terminus domain, and it required the C-terminus in order to keep it dimerized. No, this was all good, but we wanted to understand what exactly it does when it binds the DNA. So in order to do that, we took help of single molecular imaging, again with Jay Kuhn's lab. What we did here was we had a DNA spread on a glass slide, and the idea was if we give it MOC2, are we gonna see some kind of a compactions which will appear as punctase. Now, this is a very sensitive experiment, so we need really good controls. So one of the controls which we had was this N39A mutant of MOC2, which is known not to dimerize through its ATPase domain. And indeed, when we did this single molecule assay, we didn't saw any punctase in it. We then used an S87A mutant, which can bind ATP, but it cannot allow it to hydrolyze it. And again, in this case, we didn't really saw any kinds of punctase forming when we introduced MOC2 to the DNA. Then we used just the ATPase domain, and again, we didn't saw any punctase, and we also used the C-terminal domain, and again, there were no punctase forming. Excitingly, when we give it the full length wild type MOC2, we do start seeing punctase in it. So what that tells you is that first, C-terminal domain is required. It's not just the ATPase domain, which is enough to cause that compaction because that's the domain which hydrolyzes ATP. And also, it requires hydrolysis of ATP as compared to few other chromatin remodelers where the binding of ATP is actually enough. And even more excitingly, when we use our phosphodiesterase mutant, we start seeing the punctase are forming much more early. And we can actually quantify that, and we see that these chromatin remodeling events or the DNA compaction is happening twice as fast in the phosphodiesterase mutant as compared to the wild type. And if you actually map this DNA compaction activity with the ATPase activity, seems to be a very strong correlation between the how fast it hydrolyzes ATP and how fast it's going to compact the DNA. We then wanted to understand how exactly that compaction happens. So there are two models out there in the field. One is based on SMC proteins where it happens via loop extrusion, where you have the SMC proteins, like for example, the cohesin, which comes and dimerizes a certain region of the genome, and then there is an active ATPase motor, and through that, the loops are extruded and DNA compaction happens. Second one is coming from C. elegans MORC1, where one which is that kind of compaction is called loop trapping. So what happens in here is one molecule of MORC1 will come and bind certain regions of the genome, and then it calls upon other MORC1 molecules and starts trapping loops and more loops into it. So we wanted to probe like which mechanism, which mechanism does MOC2 follows. But one important note here is in MOC1, they showed that for this loop trapping based compaction to happen, you only require ATP binding, but not its hydrolysis. Whereas in our case, we have already shown I've already shown it to you in previous slides that you indeed actually require ATP hydrolysis, very similar to the way the SMC proteins require ATP hydrolysis to cause compaction. Okay, so to prove which mechanism MOC2 is using, we again took help of atomic force microscopy. And if you look at these images, these are all those different mutants, the S87A, N39A, the ATPase domain, and the C-terminal domain. And the things which you see in yellow, those are the DNA, and those little dots which I have marked with the pink arrow, those are the MOC2 molecules. And in these images, we don't really see any compaction. However, if we give it the wild type MOC2 now, we can start seeing that the DNA has indeed compacted, and we can see loop extruding like on the peripheries of this compacted DNA. So that tells you that most likely the way MOC2 compacts DNA is indeed by loop trapping. And this is not just one experiment. We have done other single molecule experiments in order to actually confirm this mechanism. So if you put all of that information together and put it back into the cells to understand like whether indeed there is an effect when we have MOC2 either in the phosphorylated state or in the dephosphorylated state. So what we observe is when we give a phosphomimetic version of MOC2 in cells where the wild type MOC2 is actually knocked out, we didn't really see much difference in the accessibility of different genes. And this is an ATAC data, by the way, which I'm showing you here. But if you look at the plot on the left, which contains the phosphodiesterase version of MOC2, then indeed there is a lot of compaction which happens in its presence. So that also shows you that this observation which we saw in and we using our single molecule assays is indeed happening in cells as well. So that leads me to propose the model. So we believe that the MORC2 comes as a dimer through its C-terminal domain, and it does a genome surveillance in its phosphorylated state. And when it gets a cue for compacting the DNA, it gets dephosphorylated and it clamps onto the DNA. And then we don't really have much evidence, but like it then calls upon other MORC2 molecules in order to cause that compaction. And all of this is actually governed by a phosphorylation based modification, which governs all this function of MORC2. So what about the other MORCs? So we first concentrated just on the MORC3, and now this is an unpublished data. So we are now able to express and purify a full-length MORC3. And we also wanted to make a comparison to its ATPase domain. And here, if we perform the DNA binding assay on the full-length MORC3, we observe that it does bind DNA, but not as strongly as as compared to the MORC2. And it can bind DNA in presence of histone peptide, the histone 3 peptide. And the reason we do it is because this MORC3 is a known binder of H3K4 trimethylation mark. Next, we also did the DNA binding in presence of the H3K4 trimethylation peptide, and we didn't really saw much difference. We then went on to perform the ATPase assays on this full-length MORC3, and interestingly, we see that the full-length MORC3 doesn't its ATPase activity is actually more dictated by the presence of the DNA as compared to the H3K4 peptide, whether it's present in its unmodified form or in the trimethylation form, which was quite interesting as compared to just the ATPase domain, for which there has been previous work showing that indeed you require the H3K trimethylation peptide and DNA in order to show ATPase activity. So what our preliminary data is showing that for the full length MORC3, the ATPase activity is indeed dependent on the DNA, but not on the H3K4 trimethylation mark. And this is my last slide. So here again, this is a very preliminary data, but what we showed that MORC3, like MORC2, can also compact DNA, but it compacts DNA only in the presence of the, it compacts DNA only in the presence of the H3K4 trimethylation, H3K4 peptide, but it doesn't really require trimethylation mark in there. And the other interesting observation is it compacts DNA at much slower rate. This is all very preliminary, so hopefully maybe I can come back next time and tell you more about the MORC3 story. With that, I would like to thank my lab. So this work was spearheaded by Vinny Tan, who did most of the MORC2 work, and then the MORC3 is now done by Mariam Sana, and she's very well supported by Rebecca. And I would also like to thank all my collaborators and the funding agencies. And thank you for listening.
Awesome. Thanks so much, Shabih. Those movies of the flexible DNA being compacted down at the puncta are really, really cool. We have time for just a few questions. So audience, if you would like to put a question into the Q&A, please go ahead and I can ask it in the next minute or two here. Or if you want to raise your hand, I can allow you to unmute quickly. Otherwise, you can keep putting questions in the Q&A once we move to the second talk, and Shabih can answer those in the Q&A box while Kazu is speaking. No one's raising their hand, so I will just ask one quick question. In your genomic analysis of MORC2 binding, like it's kind of anti-correlated with H3K9 methylation, but do you see, like, is it enriched at boundaries or places where you see a drop-off of H3K9 signaling or methylation?
No, I think it's very well away from those. It's mostly enriched in the promoter region. So the next one is the line one. There it is indeed somewhere around the boundaries. But the important thing is it's meant to be all within the H3K9 trimethylation mark regions. And
Right.
the reason it's not happening is because it hasn't really received the right cue. And the right cue for it to go to those marks is when we get retroviral infections or we get like active line one retroelements. That's where the hush gets activated, and once that's get activated, that recruits the MOC2. Whereas the experiments which we have done is in the absence of those cues. So in the next phase, we actually want to repeat these experiments while doing either a retroviral infection or something similar.
Awesome. That will be very interesting.
Yeah.
Um, well, we went a couple minutes over, so I think we'll go ahead and encourage attendees to ask questions in the Q&A if they can, and otherwise we will move on to our next speaker. So thank you very much again, Shabih, for the
Thank
wonderful
you.
talk.